The data which we present at ITOC are actually quite preliminary, and it's a presentation on SB2115, which is a STING agonist. And what is actually unique compared to most of the compounds which are developed in the space, that this compound can be actually administered intravenously, while many other compounds which are being developed in the class are actually designed for intratumor or intramuscular or subcutaneous administration. Even though immunotherapy has emerged as a really transformative approach for the treatment of cancer, there is still a significant percentage of tumors which don't really respond to immune therapy with immune checkpoint inhibitors such as CTLA-4 antibodies or PD-1 or PD-L1 antibodies. And that's actually led to development of drugs such as STING agonists, which are actually meant to conceptually increase sensitivity to immune therapy by actually induction of innate and adaptive immunity mechanisms with activation of cytotoxic T cells as well as NK cells to kind of increase the chance of the durable responses. SB2115 has been actually effectively tested in multiple preclinical models and demonstrated efficacy in colorectal cancer model as well as in melanoma and breast cancer model. And that provided enough evidence, especially taking into account that also synergy was actually shown with the PD-1 antibody, to actually take this compound to the human clinical trial, which is currently underway, and which I have a pleasure to participate in. So, as required by usual regulatory requirements, I mean, we have to test SB2115 as a single agent, which is currently going on. And in this phase, we will determine the maximum tolerated dose or recommended phase 2 dose for the single agent monotherapy. At the same time, using so-called bifurcation design with a little bit of staggering, we are actually allowed to open also the combination, which was initially designed to be SB2115 with Nivolumab, which is a PD-1 antibody, which is FDA approved in many indications, including lung cancer, melanoma, and other cancers. But due to actually ongoing collaboration between Springbank Pharmaceuticals, which is the biotech developing SB2115, and Roche, Nivolumab has been actually replaced by atezolizumab, which is a PD-L1 antibody approved for breast and other cancers at the moment. Once we complete the dose-defining phase for the both single agent and combination, there will be 3 expansion cohorts looking at melanoma, head and neck cancer, and tumors which are either naive or relapsed or refractory to PD-1 or PD-L1 antibodies, which don't fall into the cohorts which I previously mentioned. Patients who qualify for this study are patients who are in reasonable performance status with ITOC 0 to 1, who have a reasonable life expectancy of at least 3 months, have measurable disease, acceptable organ function, and acceptable cardiac, pulmonary, and other parameters. So we are currently actually enrolling into both part 1A, which is the single agent, as well as part 1B, which is the combination with atezolizumab. And so far, the experience is very preliminary, but we haven't seen any immune-mediated side effects, such as cytokine release syndrome. So the program is actually moving, despite COVID pandemic, ahead very nicely and smoothly, and hopefully will contribute to expansion of immunotherapy options for cancer patients.